So this is something that psychedelics do, and it's something that we can quantify. And I want to quantify it in zebrafish because I like zebrafish. Zebrafish, if you think they're less like people than rodents, you're not entirely wrong, but they are 1/1000th the cost of a rodent. So you can do a lot of tests and you get a lot of bang for your buck. So like one mating pair, you get a large clutch of 200 eggs that fits in one petri dish. If you wanted 200 rats, you need 600 square meters, something like that, of lab space. So you can get a lot more done. And they are fertilized externally, which means if you want to do any kind of genetic modification to the fish, which I like to do, it's really easy. It's a lot more difficult than a mammal. And they develop really rapidly. So within 48 hours of fertilization, it'll be a free hatched and swimming individual. It's going to have all the major organ systems in place, different neuron subtypes. It'll have a liver, so you can measure what kind of metabolism is happening in your drugs. It'll have a blood-brain barrier, so you can see what drugs are getting through that. And there's just a lot of benefits to dealing with the whole animal versus just looking at cells in a plate. And it's even cheaper than looking at cells in a plate. They respire aquatically, which is not great for modeling lung disease, but it is great for getting drugs into them because you just have to drop the drugs into the wells in the plate, which they fit in. They're only 2 millimeters long when they're first hatched, and they'll absorb them through their gills. And the other amazing thing is that they're transparent, which allows you to visualize a lot of really interesting and clinically viable information in live animals. So on the lower left here, you're looking at blood cells going through a heart. That's a fancy one, but you don't need a fancy fish to look at that. Oh yeah, this is the system we're using to look at them. It's a modified version of this bioimager, and what it does, it automatically sucks up the fish out of the 96 well plates, transfers it to this optically clear capillary where it can automatically rotate it and zoom into the cellular level to automatically image it, the parts you're interested in, really reproducibly, which is great. But yeah, as I was saying, you don't need a fancy fish to look at the heartbeat. They're transparent. You can just, we can look at one fish a minute and see if the drugs we're giving them cause cardiac arrhythmia or other cardiac effects or not. We do need slightly fancier fish, these genetically modified fish that are expressing fluorophores from jellyfish and coral. And the first three lines we're looking at are looking at the specific neuron subtypes that are targets of ibogaine. And that's how we're going to see that it's these different molecules are still active in the same neuron systems that we were looking at before. There are a lot of different metrics you can use to quantify neuroplasticity. We can look at the top 5 all at once, but excitability, that a neuron will fire again once it's received some stimuli. We need another special fish for that. This special fish has calcium-sensitive fluorophores, so that what you're looking at here, this is the shadow of action potentials. So these are neurons firing in real time in a live animal, and we can use that and differentiate when we're seeing, we can quantify the agonist activity of the new drugs, and we can quantify whether the reaction is likely agonist or reuptake inhibition, and we'll do that in all the same subpopulations of neurons. And then the other ones, they can all be quantified at once with a more stable fluorophore. Again, these are live fish. This is one I took. This is the dopaminergic neurons, and we can count them. And so if we were looking at a brain injury, we can see how the neurons might repopulate and if they might be improved repopulation around where that brain injury is. And how to give them a brain injury, there's lots of ways, but I think the best is just turn your microscope laser up to 11 and blast it. Here, we're just blasting one neuron, but we're probably going to be doing a dozen or so. We can do this in a very controlled and reproducible way because it's the focal volume that's being blasted. And so I'll just end this by walking through that experiment. They get automatically arrayed into the 96 well plates, drawn into the capillary. It'll be focused to a reference image. So you'll be focusing in on the exact same spot, and then you do that very reproducible laser ablation to knock out the number of neurons you want to knock out. Put it back into the plate, and at that point, you can dose it with the therapeutics in a dose-dependent manner. Give it a day to recover, grow some new neurons, and day 2, quantify any beneficial effects you might see around those neurons. And we can do this all at once, and we're left with ex vivo data too, to look at things that pharmaceutical companies are interested in: toxicity, other imaging, metabolites. Yeah, and we can do that. So most of these experiments we can do at once, and we can use all this data to train the next iteration of new compounds to look at. And I'll leave it there.